All right, very good evening, everyone. All the students and participants of MOOC course Introduction to Proteomics and Interactomics. Welcome to this live session. Today we are planning to actually show you two-dimensional differential electrophoresis or 2D DIGE technique, how it works. And also we plan to show you microarrays, especially protein microarrays, how that experiment can be done. Okay. All right, so in the last week live session, we actually showed you the traditional two-dimensional gel electrophoresis. There are different steps involved in doing 2D based proteomics workflow. After doing the protein extraction, then what all steps you have to do, that we showed last time. I'll just kind of quickly recap you that the steps involved, starting from rehydration of the strips. So we have the trays here, and this is the IPG strip. So ideally, you want to take one of the IPG strip, which can be 3 to 10 pH or 4 to 7 pH. Then you will take your protein solution and you will place it inside this rehydration tray. Then you are going to place your IPG strip so that protein can be rehydrated and getting immobilized inside the IPG strip. The next step is that after doing the rehydration, which is a passive rehydration, then we are going to do the second step, which will be isoelectric focusing, and where we can use another instrument setup, which is IPG4, which is this instrument where we can place this tray here, and IPG strips could be placed here, and we can do the isoelectric focusing, which is ideally the first dimension separation of proteins, right? So once we have done the IEF, the first dimension protein separation, then we can take out the strip and we prepare them for the second dimension by the process of equilibration. And once we have done equilibration, then we are ready to do the second dimension separation, which we showed you this type of the large gel setup. It had 6 units here, which we have for running 6 large gels, right? So if I just show you the cassette here, and here you can place the gel, right? So ideally, this is what involves for doing any kind of 2D gel based proteomics workflow. Now let's move on and then say that if there are 2 gels you're running and they're not so reproducible because of the running artifacts, because of variability in them, so then our intention is to reduce that variability, and that's where difference in electrophoresis has really proved very useful for doing quantitative proteomics using gel based methods. So today what we plan to do, we are going to show you the labeling strategies, how to label the protein using Psi3, Psi5 dyes, and also you make an internal pool from both your control and the test conditions, where then you can label those samples with the Psi2 dye, the third dye. Just imagine that you had 5 control samples and 5 treatment samples, and now you want to compare those on the gels. So you are going to make a pool of all these 5 controls and pool of all 5 treatments, mix all of them together, and that you are going to use as the internal standard. That internal standard you are going to label separately with the Psi2 dyes, and then you will have controls and the treatment samples which you can label with Psi3 or Psi5, and ideally you can swap. Some samples are having Psi3 of control, and some of the controls are having Psi5 dyes. So ideally, this is the experimental strategy which you need to follow for doing DIGE experiments. But actually, what you do for doing the labeling, this is what TAs are going to demonstrate you today in the wet lab experiment, that how can you do the labeling. After doing the labeling, then you can scan the gel using one of the sensitive scanner, which we have, a Typhoon scanner, where you can use multiple wavelength, and same gel will produce different images for Psi2, Psi3, and Psi5. And then if you have multiple gels, then again Psi2 images from each of the gel can be used for the normalization purpose. So these are some of the steps which we are going to talk to you today in the first part of this live demo session. The second part, which is let's say around the next 15 minutes or so, afterwards we will have a session for the microarrays, protein microarray technology. Because we are doing the labeling of the proteins, which is labeling with the Psi3 and Psi5 dyes, and when microarray technology is also linked to the same dyes, so we thought to club the two of these technologies together, and irrespective of which courses you are taking, I think these techniques are very useful for you to know and understand them. So the second part, we are going to talk to you about protein microarrays, how to do the assays using the protein chips, what are the steps of doing the incubation with primary and secondary antibodies, how to scan the gel image, in fact the microarray slides in that case, and then finally you can place the grid on the microarray chip, and that can be used for the data analysis part. So the second part on the microarray will focus today on doing data analysis parts and the scanning part, where the first part is going to show you the critical step of doing the protein labeling. So let me now invite the TAs here. I have with me Vipin and Kanika. They are the teaching assistant for the first part, and then I have with me here Nikita, who is going to be showing you the protein microarray part, and they are going to show you some crucial steps. Whereas some of the things we have to really skip, and we have to just tell you theoretically what can be done for these experiments. Because of the nature of the experiment, it takes very long time, but at least we can give you the critical ideas for some few steps involved in DIGE as well as protein microarrays. So let's first start with the DIGE technology and let Kanika take over.
We welcome the participants from both the courses, Introduction to Proteomics and Interactomics course. So let's just get started with the DIGE techniques demonstration. So DIGE technique, as you all know, is an advancement of the traditional 2D technique. So in 2D technique, what we generally do is, if suppose we have two samples, then we run two different gels and then we manually do the analysis and annotate the gels. But in case of DIGE, what happens is, we can run a single gel, we can label the two samples, we can run them on a one common gel, and then which will significantly reduce any gel to gel variations. Additionally, we can also incorporate internal standard, which is nothing but a mixture of both our control and the treatment sample, so that again the sample variations will be reduced drastically. So DIGE workflow is basically developed by GE Healthcare, and it involves three different dyes. So labeling is by Psi2, Psi3 and Psi5. These are the three dyes, and they make a covalent bond with the epsilon lysine group of the protein. So it has an hydroxy group, hydroxysuccinimidal group, which covalently binds to the lysine group. Lysine also has a +1 charge, and so does these dyes. So overall, the charge of the protein remains same. The sensitivity of psi dyes is extremely good, equivalent to the silver staining. So we'll just show you how do we do how do we carry out dyes in our day-to-day lab experiments. So the workflow is more or less similar to 2D, but what happens here is, in 2D we first do the rehydration of our strip and we add our sample there. But in this case, our sample preparation is a little bit more complex than the regular 2D. So what we take is, we take equal amount of samples. Suppose in case we have control and a treatment sample, we take equal amount of samples, for example 60 microgram from both the conditions, and then we label with the different dyes. So suppose we have already taken 60 microgram from control and we are adding Psi3 dye to the control. So we'll take the dye volume, we'll add to our sample which has our control sample. So we have already added our dye. Then in next case what we do, we take the next dye Psi5. So Psi5 again we'll take, and then this will add to our treated sample. So sometimes one can also do dye swapping. So in this case we have taken control, we have labeled with Psi3 and our treated sample with Psi5. One can swap the dyes. We can use Psi5 for our control and Psi3 for our treated sample as well. Then in case of Psi2, what we do, we take equal amount of Psi of the control and treated sample. For example, we'll take 30 microgram from each, we'll add in a common tube. We'll take one tube which has both the samples, and then we'll add Psi2 to that. So the sample in which Psi2 is there has both control and treated. So that's why it is like an internal standard, equal amount of both the samples. Now the total amount of protein in all the three tubes is same, whether it's labeled with Psi2, Psi3 and Psi5.
Now we have already mixed, like labeled our samples, so we put for 1 hour incubation, so that like whatever the samples will be labeled by our dye. So once we put the like 1 hour incubation, then we have to quench the reaction. So basically we have to stop the reaction. So in this case, we add the 2 microliter of lysine. So this we have already made lysine. I'll take 2 microliter of lysine, then I'll add in each sample, like in cyto. Similarly, I will add for in site 3. Yeah. So now after adding the lysine, we'll wait for 10 to 15 minutes so that like all of the like reactions have been stopped. So once it will be done, so the next step will be like mixing of all sample. Like we have a sample which is labeled by site 2, sample is which is labeled by site 3 and the site 5. So once like 15 to 20 minutes will be there finished. So we'll mix all samples in one tube.
So once we prepare our common pool, we go for the rehydration of IPG strip. So this is a protein sample which has pool of all three labeled dyes, and then we'll add a rehydration buffer to this sample pool. After we have added the rehydration buffer, then we do our regular IPG strip rehydration, which is an overnight step.
So the time make the volume makeup till
So you can
350
see
microns.
this rehydration buffer. We've also added bromophenol blue, so its
Yes.
overall color is blue. So we'll also vortex it for a brief time. Now our protein sample has been prepared, and we will first add our protein sample in the tray. This is the same step which we had done in 2D as well. So first we'll add the sample. We'll make sure that there are no bubbles here. It's all uniform. So amount of the protein and the total volume depends on the strip length. So once we have added this, we'll take our IPG strips. IPG strips, as we all know, are commercially available with a plastic coating on top of them so that they don't get disrupted. So we'll first peel off the plastic film. So our gel side is exposed. Now we'll keep the gel side in a way that it is in direct contact with our sample. So gel side is downwards, and we'll mix it well so that no bubbles are there. It's a uniform distribution throughout the strip. So this is an overnight step. Rehydration usually takes at least 16 hours or so. So once rehydration is done, the next step is separation in the first dimension using isoelectric focusing. So this is our unit. You can see there is a gel tray here. Then we have the two terminals, positive and negative. You will see same positive and negative terminals on your gel strip. So we'll place our strip in that manner that positive end is towards the positive terminal and so. So isoelectric focusing, again, it takes around 16 hours or so, 16 to 18 hours, and the proteins, they get separated on the basis of their PI values. So once isoelectric focusing has been done, we do equilibration of the strip just to prepare it for its separation in second dimension. So after equilibration, we do second dimension base separation. It's an SDS page gel. You can see this is the cassette. It's a big cassette. So we have already added a glass plate. On top of the glass plates, we keep our strip, and then we'll put this cassette into the big electrophoretic unit. The run itself again takes 3 to 4 hours. Now, the only, again, another difference in 2D and dyes is that in case of 2D, we have to stain our gel with Kumasi or any other dye. But in case of dyes, we do not have to do any staining or destaining because our dyes, we do our detection based on our dyes, which are fluorescent in nature. So, after we get the gel, after the separation has been done in second dimension, we scan the gel at different wavelengths. So these are the different scanning parameters. So side 2, side 3 and side 5 we use 3 different wavelengths. For side 2 we use around 473, for side 5 it's 530, for side 3 sorry, and for side 5 it's around 620. So we'll show you a representative gel image of a 2D dye gel. So this is how a 2D dye gel looks like. So you can see on the right hand side the first gel which is in blue in color, so that it's the same gel. All these 4 images are of the same gel. If we see the first image, in this the scanning has been done at only one particular wavelength which corresponds to side 2 dye. So you can see it's blue in color. So side 2 has our internal standard, as you may remember. Next gel, it has been scanned at a wavelength which is for side 3 dye, and our control sample has been labeled with side 3. The last gel, it has been scanned at a wavelength which corresponds to side 5, and side 5 was used to label our treated samples. So you can see just on, like, visually you can see, like, you can there are brighter protein spots in the treated sample which was labeled with side 5 as compared to side 3. So like that we do our gel analysis, and the image on the left hand side is usually when we acquire a combined image which has been scanned at side 3 and side 5 wavelength. So again, as we have mentioned, the same gel has been scanned at 3 different wavelengths. This is for side 2, this is for side 3, and this is for side 5. So looks like we are done with the demonstration of the DIGE technique. Now we'll move to the microarray technology. So Nikita will take over the demonstration of DIGE technology, of microarray technique.
You can start this.
Thank you, Kanika, for the introduction. We will be now looking into the microarray technique. Here, I will be giving you a brief overview of how protein microarrays perform, and then further we will be looking into basic steps and how the scanning is done, and how the further steps of grid laying and the data is acquired. So, to begin with, we will be dealing with the autoantibody response in protein microarrays. So, what are autoantibodies? Autoantibodies are the antibodies which are produced. Autoantibodies are the antibodies which are produced against our own proteins. It is majorly reported in autoimmune diseases, but a different type of cancers have also shown autoantibody response. It is majorly because overexpression of the gene or altered proteins, or due to different subcellular localization of the protein, or probably because of the post-translational modification. In all these cases, the body takes your own protein as an antigen, and then it creates an autoantibody against it. So we are basically interested in a study how autoantibody response is studied in cancer, and so for that, what we have taken is we have taken a patient cohort and a control cohort, and we have incubated the serum or CSF of these proteins of these samples on the Uprot chips, which contains 19,000 protein spots and duplicates. So the workflow for the protein protein microarray for autoantibody screening is very similar to that of Western blotting, where we incubate the serum or CSF of this patient or a control patient onto this chip, which contains protein spots and duplicates, and which contains recombinant proteins. And these proteins are the chip that we are using here contains the contains the GST tag along with the protein. So what we do is we incubate, we first block the slide, and then we first we we first block the slide, and then what we do is we incubate it with primary antibody. For primary antibody, we use serum, which contains the antibody against the protein of interest, and the anti-GST antibody, because that is the tag which is associated with the protein. And then it will go and bind, anti-GST will go and bind throughout the slide, whereas whereas anti-serum, and whereas the serum antibodies present in the serum should only bind to the should only bind to the proteins where the autoantibodies have, for which the antibodies have been developed. Further, we do a secondary antibody incubation where we use anti-human antibody and anti-goat, because the anti-GST is produced in goat. So anti-goat, anti-GST antibody, and then this antibody, the anti-human antibody is labeled with Sci5, which gives a absorbance at red wavelength, whereas anti-goat antibody binds to the anti-GST antibody and gives a green fluorescence. So the scanning is performed at dual channel. We will just give a brief overview how the experiment is performed. We can look into these steps for that. So if you see, this is how the protein array looks like. So this is the barcode region, which do not contain the protein. This area contains the proteins, which contains proteins in imprinted, and you always touch the slide. You always touch the slide from the barcode area and not the other area, because it can lead to imprinting of your fingerprints. So here, what we do is, so firstly, we take the slide and then we thaw it, and then we put it for blocking. So this is how the blocking looks like, and this is the blocking solution. So now we just pour it over here, and then we do a gentle rocking for 2 hours, and the slide will be incubated. Same incubation steps are performed for primary incubation, secondary incubation, and blocking. After blocking, we remove the excess blocking solution or primary or secondary incubation. What we do is, we remove the slide and we put it for washing. So always make sure that you touch the slide from the barcode region, and then this is the cassette.
So this is the magnetic bead. This magnetic bead allows the proper circulation of water. And after, when we change the wash, what we do is, we just take out this slide and we switch these positions so that the sides are all uniformly. Then we place it back in the socket, and then we allow again to wash this. And each wash of 10 minutes are given 3 times consecutively for washing the extra toxic solution, primary antibody solution, or secondary antibody solution. Once the experiment is performed, we do a scanning of the slide.
Okay, so
what you may realize, it looks very simple for doing microarray experiment. Just the last slide has to be watched many times. But you know, a lot of the wet lab risk is actually being shown while you are doing this kind of washing step, which has to be very rigorous. You have to make sure that your gloves are not touched, and you have to use very good forceps, and you have to do very clean washing. At the same time, you do not want any of the protein content which are printed on the chip to really wash off. So delicate balance has to be made of rigorous washing several times, as well as not so much that it is going to remove your protein content printed on the chip. So, but this has to be done many times, which is what Devendra is telling you right now. And once you have done the whole day of steps like this, of incubating with the primary and secondary antibodies and washing of the chips, then only you are ready actually for the final thing to see, on the scanner that how the chip looks.
So once the secondary incubation is done, which is majorly done on the in the lab, then we wash the slides and we dry it, and then we put it on the scanner. And this is how the scanner looks like. This is the scanner area, and this is the area where we keep the slide. Slides are kept majorly inverted position. That is, the slide should be facing the back side. The protein printed area should be facing downside, and then we close the slide, and then we allow it to scan. Now I will be giving you a demo of how the scanning is performed.
One more. It's not shared thing.
So now there's a software called as GenePix Pro software. You can click on this software and it will search for the scanner. The slides we have already kept inside the scanner. So now what happens is you can see that there are dual channel wavelength, that is 635 and 532. You can set that, and then pixel size is kept as 10. And then now you can also change the PMT settings. It majorly defines how much absorbance it should show. So as we go down through the scanning, we will see how PMT settings affect the slides. So let's just start the preview scan. Slides is already kept inside the scanner. So this is the button for the preview scan. So we'll just start the scan. So preview scan is just like a normal scan, and we have to select the area for scanning. So here you can see that there are some spots which are coming, and if you zoom it, you can see that these are the protein spots coming onto the slide. And as you change the PMT settings for these, you can see how drastic these changes. So let me just put this setting as 450. You can see that there is a difference in the scanning, and now if I reduce it to 200, you can see how it affects the scanning parameters. So we will just keep it back at 350 again. So this is how the PMT gain affects the scanning of the slide. So and we have to be sure that we don't want most of the protein spots to be saturated, and then we don't even want to lose a lot of protein spots. So we have to strike a very good balance between what PMT settings should be done. So there is another option called as Auto PMT. If we select this Auto PMT, the instrument itself scans at 3 different wavelengths, and then it selects the best of the scan settings at which most of the spots are not saturated, but then we don't lose the spots, and then we can get a specific PMT settings at which the slide should be scanned. But
one thing should be considered. For doing one set of experiments, all the PMT settings should be same across the experiments. Otherwise, it could be an experimental error. So once that is done, once you scan the slide, what you do is you save the, this was a preview scan, the scan area. So here if you see, this block is the scan area, and then you can just demodify it to the length you want it to be. Make sure that you don't lose on any of the protein and the scan area is set properly. And then you can start the scanning of the slide. So because this will take a lot of time, we are not going to do a proper scanning of the slide, but I'll just show you how the further processing of the microarray slides are done. And then you can see that now the scanning has started for the selected area. Now I'll just show you how the analysis is performed. So then we go to the GenePix Pro analysis only software, where we can open the image that was saved. So now you can see the image, how it looks like. This is for the green channel data, and then you can just zoom into it. This is how the scan slide looks. And if you go to the features, here you will not see any name, because right now these are just protein spots without any identification. So then each and every vendor that provides you slide will also provide you with a GAL file which tells you exact location of the slide of the proteins which are present on the array. And then we have to overlay that grid over it. So for that, what we do is we'll just go and load array list. So this is how the grid is laid. You can just zoom it to arrange it properly. And then you can just so now you can just see, when you go to the protein spot, you can see that there is a name for the protein, and then you can just select the block. And then you can just try to impose the grid properly. Now you see that only for this block, the grid has been laid. And then when you zoom into it, you can see that the software has manually tried to pick up these spots. So here, this is the setting feature where you can tell what is the minimum diameter of the spot and what is the maximum diameter of the spot, so that it selects that spot area only and not the artifact like the background or a very small specks. So we can also tell what is the CPI, that is the background intensity and the foreground intensity, so that it tries to find only the spots and not the background. Like here, if you see, it has actually taken the background as a spot and not the actual spot. So what we do is we just select the feature and we try to resize it, and then we try to resize it. And this is how the resizing of the features are performed. Similarly, if you look into this area, this spot, you can just select this spot and try to resize it.
because it has taken the background into consideration. And then once that is done for all the slides, you can just select the data, and it will give you foreground intensity and background intensity for all the slides and all the features at 635 nanometers and at 532 nanometers. That is at green channel and red channel. And then, as you see, this is a GAL file, and this is how it looks like. The 532 median and 532 mean, and there's a standard deviation for that. This is the green channel data. Similar data you will also obtain for the red channel. Red channel data will specifically specify that what all autoantibodies are present against which proteins, and then you can actually do a statistical analysis across different slides, and then you can compare which all proteins are showing an autoantibody response. Which all proteins are showing the autoantibody response against the protein. Then you can just select this area, and then you can save the results. As .gpr files. Very system hang okay. So now this is for one of the studies where we can see that further once we obtain this thing, once we obtain the results for the red channel data, we do a normalization, and there are certain control spots, that is positive control spots, which contain GST and IgGs, which should definitely show a fluorescence at 532 for the green channel and 635 for the red channel at the red channel. And then further we do intra-chip reproducibility to check how the slides have been processed, and then we also do an inter-chip reproducibility to check how the background throughout the slide has been performed properly. So there are certain control spots, like positive control spots which contains IgGs, and negative control spots which only contains blank or have nothing imprinted onto it, which is usually used for the to check the background of the slide, overall background of the slides. Once we have all the data for the for 635 and 532 channel, then we do a normalization to remove the inter-chip variability and day-to-day variation. Once that is performed, then we perform the analysis group. Then we try to group the samples, and we try to look into the proteins that are showing differential protein response. So here are some of the examples where you can see that some proteins in control have shown no response in the red channel, that is autoantibody, whereas in the grade 4 patients of glioma, they have shown a good autoantibody response. And then this is another protein. Similarly, you can perform an autoantibody response, and we can also perform certain functional assays to look into how proteins are activating or acting in a disease condition as compared to normal condition. So this was all about the protein array. So this was all about the protein arrays, and I hope that this session for DIGE and protein arrays was helpful, and the link for the video will be shared to you, so that if you have missed onto some of the details, you can go later and check it and visit the link.
So thank you participants for being with us throughout the demonstration. We hope that you really liked it. So in case you have any queries, you can please post it to us. We'll try to answer as quickly as possible. Thank you.